Hi, everybody. Today we are going to talk about the process of meiosis. Meiosis is a type of reproduction. What's involved with the type of reproduction called sexual reproduction, which is different from mitosis, which is asexual reproduction. So the question that shows here, how do you distinguish between sexual and asexual reproduction? Well, with asexual reproduction, we are making clones. So it's fast. Asexual reproduction is usually easy. It doesn't cost too much energy. So unicellular organisms like bacteria might clone themselves or asexually reproduce. A hydra, which is an aquatic organism, might have a little clone bud off of itself, or like many plants, they can actually just clone themselves. Sexual reproduction usually involves two parents who will have two cells that fuse to form a new organism that is slightly different from either parent. Sexual reproduction is usually more energy intensive, but it does have other benefits, which we will get to in a moment. So, meiosis and sexual reproduction. Mitosis is the same thing as cloning. Remember that mitosis produces cells with the same information, so identical cells, which are exact copies. So they are clones with the same amount of DNA, the same number of chromosomes, and the exact same genetic information on each of the chromosomes. So when we undergo mitosis, we are cloning our cells, right? Which is why every skin cell you have should be the exact same, because you're really just cloning them. So single-celled eukaryotes like yeast, which is a fungus, certain protists, they reproduce asexually. Remember, it is fast and it's easy and it doesn't cost too much energy. The hydra can do it as well. All right. So what are the penguin is asking, what are the disadvantages of asexual reproduction? And then it's asking, what are the advantages? So the advantages I've mentioned several times now. It's fast, it's easy, it doesn't cost too much energy. But there are some disadvantages. What do you think some disadvantages might be? Well, some disadvantages might be that when you create clones, every single of your offspring are going to be the exact same. There's no variation genetically. So if you had an entire population of clones, and let's say a disease came through, well, if every single organism in the population is a clone and a disease comes through, what's going to happen? Well, they're all going to respond in the same way. And what if that response is by dying from the disease? Then every single organism is going to die and your population would collapse. So that would not be a good thing for a population. So asexual reproduction means that you're more susceptible to environmental problems like a disease or temperature change or increased rainfall or a pH change or all sorts of things. Sexual reproduction has some advantages. And its main advantage is that we are making genetically unique offspring. And so that means that we won't all respond in the same way when there is something like a disease or some other environmental change. It increases the likelihood that the population will survive. All right, now there are disadvantages that sexual reproduction takes a lot of energy, right? There's a lot of energy behind it, but the advantages seem to outweigh the disadvantages. Sexual reproduction takes advantage of mutations. Mutations can be either neutral or harmful, and very rarely, every once in a while, there are good mutations that can help an organism. Mutations are perpetuated by DNA replication, and unless they are lethal, mutations are passed to offspring and become part of the genetic makeup of each species. When we undergo sexual reproduction, we reshuffle genes, and that may combine different alleles in new and possibly beneficial ways. All right, so genetic variation is the way that evolution basically starts. Some individuals are better suited to an environment, other individuals are not so well suited to an environment, and the ones that are better suited usually survive and pass on their genes. And that's the basis of natural selection. Okay, so for sexual reproduction, what if a complex, like an animal or a plant, multicellular organism, like humans, want to reproduce? Well, in order to do that, we need to join an egg cell and a sperm cell. So the question is, can we make egg cells and sperm cells by the process of mitosis? And the answer to that question is no. All right, and why is the answer to that question no? Well, let's think about it. Here's an egg cell, and as we already know, human cells have 46 chromosomes. And here's a sperm cell, and human cells already have 46 chromosomes. So if we made egg cells and sperm cells through the process of mitosis, they would each have 46 chromosomes. And when a sperm cell fertilized an egg, we would end up with 92 chromosomes, and that's way too many. A zygote does not need all of those extra chromosomes, and they in fact may be harmful. So this will not work. We can't make egg cells and sperm cells through mitosis. Mitosis doesn't work to make egg cells and sperm cells. So no, not the way that it works. All right, so how is it going to work? Well, to help us understand how it works, we're going to look at a karyotype. And you may remember that a karyotype is a picture of all of an organism's chromosomes that have been dyed and lined up in homologous pairs. So this is 46 chromosomes total. That means there's 23 homologous pairs. Every chromosome has a pair. So here is pair 1, here is pair 2, and so on. This organism is a human, and it's a female because there are 2 X chromosomes. We can see them there. They've been circled in red. So this is the karyotype for a human female. Now we can take another karyotype, reorganize it, and I've got to move this out of the way. And we can see that, once again, we have 46 chromosomes. That's 23 pairs. And we have an X and a Y. So that is a male karyotype with an X and a Y chromosome. So all of our chromosomes are paired. Homologous chromosomes have matching genes. Those are, remember, similar but not identical. And those genes control the same inherited characters. So homologous refers to the same information. So here is my cell with chromosomes inside of it. This cell is diploid. Okay, that's a new term. What does diploid mean? Diploid just means that all of our chromosomes are paired. We have paired chromosomes, diploid chromosomes. Our diploid cells have their chromosomes in pairs of 2. So it's always going to be an even number. Sometimes we write diploid as 2n. So in this case, my diploid number is 4. So this organism has 4 chromosomes total. So my diploid number, that's 2n, equals 4. 2 pairs gives us 4 chromosomes total. All right, so these are single-stranded homologous chromosomes, and if we duplicate them, we have double-stranded homologous chromosomes that are now duplicated. Okay, so now that we know a little bit about the chromosomes inside the cell, how do we actually make sperm cells and egg cells? Well, we have to start by looking at all of our body cells. Our body cells have 46 chromosomes, but that won't work, as we already mentioned, for sperm cells and egg cells. We need 23. So we have to reduce the number of chromosomes by half. That is what the process of meiosis is going to do. It's going to reduce the number of chromosomes by half to 23. An egg cell should have 23 chromosomes, and a sperm cell should have 23 chromosomes. And we call these types of cells gametes. You see that at the bottom there. So meiosis creates gametes, and those gametes have half the number of chromosomes. And then eventually what's going to happen, a sperm cell is going to fertilize an egg cell, and the process of fertilization will give us a brand new organism, a totally new individual that has a chromosome number of 46. So meiosis is how we solve the problem that we mentioned earlier in the lecture. How do we get a newly formed cell with 46 chromosomes? We call that new cell a zygote. All right. So, in order for meiosis to work, the chromosome number must be reduced from a diploid cell to a half or haploid cell. We see that happening in this picture. We have males and a male and a female here in very specific organs in each of these people, in either the testes or the ovaries. Meiosis will occur, halving the number of chromosomes to make a haploid sperm cell with 23 chromosomes and a haploid egg cell with 23 chromosomes. At some point in the future, a sperm cell may fertilize an egg cell, doubling the chromosome number back to 46. Here is our diploid zygote. The diploid zygote is going to grow and develop into an adult one day, and the entire process is going to happen again. So we have to have meiosis to allow this process to continue. So taking the chromosome number from 46 to 23, meiosis reduces the chromosome number to make gametes, and fertilization restores the chromosome number. All right, so there's 2 stages. Okay, so gametes are haploid, which means that they do not have chromosomes in pairs. They have chromosomes that are unpaired. So it's not 2N because there are no pairs, it's just 1N, one copy. There's no homologous pairs. After fertilization, now we have pairs, right? So the egg cell has one part of chromosome pair 1, and the sperm cell has the other part of chromosome pair 1. When fertilization happens, we end up with a homologous pair. So now we have a diploid zygote, and the diploid zygote will eventually undergo meiosis once again. And if this is a male, meiosis is going to give us sperm cells that are back to haploid. But notice each sperm cell has a different mix of the original chromosomes, right? Things got a little jumbled up there during the zygote and adult stage. So it is not, the sperm cells are not the same as one another, and the sperm cells are not the same as the dad. All right, so meiosis is going to mix things up. So meiosis is reduction division. It is special cell division specifically for sexual reproduction, and it reduces things from 2N just to N, or 1N. Diploid cells end up becoming haploid cells, and you see that happening there. It can only happen in testes or ovaries. Okay, meiosis is going to make gametes. And meiosis evolved from mitosis. So the stages are very similar, but the overall result is much different. So as we go through meiosis, don't confuse the two. Again, meiosis evolved from mitosis, so they're going to be similar, but they're not going to be quite the same. Meiosis means to diminish. It produces gametes, it reduces the number of chromosomes, and we're going to see a number of similarities to mitosis. However, during mitotic cell division, the cell undergoes one round of DNA replication and then 2 divisions. We have meiosis 1 and meiosis 2. Here's what it looks like. This is meiosis 1. It actually probably looks pretty familiar to you. Interphase, prophase 1 because it's meiosis 1, metaphase 1 because it's meiosis 1, anaphase 1, telophase 1, and then we have meiosis 2, which has prophase 2, metaphase 2, anaphase 2, and telophase 2. All right, it's like the stages of mitosis done twice with a few differences. One thing that you should be able to do is to compare meiosis 1 to meiosis 2. Meiosis 1 is the most different from mitosis. And that is because the point of meiosis 1 is to separate the homologous pairs. The homologous pairs will get separated in meiosis 1. And in meiosis 2, we're actually going to separate the sister chromatids because the homologous pairs have already been separated. So meiosis 2 is actually going to be more similar to mitosis, and we'll look at that. All right. So we are going to have DNA replication in interphase. So we see these unreplicated chromosomes becoming replicated. And then you'll notice that we have our first division. Notice what's happening here is that we had a homologous pair up here, and the homologous pair ended up getting separated. One part of the pair in one cell, another part of the pair in the other cell. All right, and then we have our second division, or meiosis 2, which is then going to separate the sister chromatids in each cell. All right, so the sister chromatids will get placed into different cells. That is the second cell division. All right. So the first step is to duplicate DNA. We already know about this, right? This is just the S phase of interphase. So why are we bothering to do it? Kind of seems pointless, doesn't it? If we're going to have 2 rounds of meiosis, why are we bothering to replicate the DNA? It doesn't seem to really have a purpose, but the reason why is because meiosis evolved after mitosis, and it was already there, so they didn't get rid of it. It's because we kind of reused this process. So it was convenient to use the machinery of mitosis. The DNA replicated in the S phase. Just like mitosis, the DNA replication is no different. All right, in meiosis 1, the first division of meiosis separates the homologous pairs. So we are going to see that in prophase 1, the homologous pairs are actually going to find each other. Does that happen in mitosis? No, it does not happen in mitosis. The homologous pairs do not need to find each other in mitosis, but in meiosis they do. When they find each other, they form what is called a tetrad. A tetrad is when a homologous pair will join together. It's sort of like they're holding hands, right? There's actually going to be a little protein that connects them here and here. This process is called synapsis. Okay, in metaphase 1, instead of making a single file line, these homologous pairs just line up. So it's sort of like a double line. They cannot make a single file line because they formed a tetrad. So the tetrads themselves make the line. So you can imagine the metaphase line is right here. And then in telophase 1, what we end up with is we end up with 2 cells. And as you can see, are the cells identical? The answer is no, they are not identical. They're different. And how many chromosomes do these cells have? Each one has 2 chromosomes. How many did we start with? We started with 4, and now we have 2. We went from a diploid cell to a haploid cell. So we've already made the cells haploid. So we've already reduced. All right. In meiosis 2, things are going to be a little bit different. So in prophase 2, there's really not much that happens. The chromosomes are there. They don't have pairs. They can't pair up because the pairs are in separate cells, aren't they? In metaphase 2, they make a single file line. And then the sister chromatids are separated. And so now we have 4 cells. And each cell is still haploid. It was haploid before, and it's haploid now. They're just, the chromosomes are not replicated. So we end up with 4 cells, and the cells are not necessarily identical, are they? Right? We have the little chromosome is blue, but here the little chromosome came from the mom. So this is the second division. Meiosis 2 seems to look a lot more like mitosis. And there's our 4 cells, and if this is a male, these 4 cells will become sperm cells. All right, so meiosis 1 is made up of interphase, prophase 1, metaphase 1, anaphase 1, followed by telophase 1. The first division separates homologous pairs. That's the key point. Meiosis 2 is made of prophase 2, metaphase 2, anaphase 2, telophase 2, and meiosis 2 is going to separate sister chromatids, just like mitosis. So here we can see some actual pictures, prophase 1, metaphase 1, all lined up as pairs though. Anaphase 1, homologous pairs are separated. And then we have metaphase 2. Notice that it's happening twice. And notice that anaphase 2 is happening twice, sister chromatids being separated. And now we have 4 cells all haploid at this point. All right, so some important things happen during meiosis 1, and we want to talk about those important things now. The first important thing is called crossing over, which takes place during prophase 1. During crossing over, when the homologous pairs form a tetrad, little bits of DNA are actually going to switch. You see that here and there, right? Like the appendages of the chromosomes parts of it will break off and they will switch with the homologous pair. This is going to create some incredible genetic variation, right? Because you have mostly the dad's chromosome, but now it has pieces of the mom's chromosome on it. Remember, it's the same genes, but slightly different variations of those genes. Notice that crossing over can't happen on the outside chromosome, or the outside chromatid, because they're not in contact with one another. So we end up, when we form a tetrad, swapping pieces of DNA. The DNA breaks off and reattaches. All right, so crossing over has 3 steps. First, we have crossing over, so the appendages come into contact. The DNA has to break, and then it has to be reattached. All right, so the penguin is asking a good question. What are some advantages of crossing over? You may have said that some advantages include genetic variation, and we know that genetic variation is going to make a population stronger. We get a new combination of traits. All right, so one thing that you should be able to do is to compare mitosis and meiosis. So you can use this diagram to compare them. Mitosis has 1 division, meiosis has 2. Meiosis creates identical cells. Meiosis creates completely different cells. Meiosis produces 2, and mitosis produces 2. Meiosis produces 4. Mitosis keeps cells diploid. Meiosis makes diploid cells haploid. Mitosis is for growth and repair, whereas meiosis is to produce gametes. Meiosis does not have any crossing over, and meiosis does have crossing over. All right. And here we see a review of meiosis 1. We have interphase, we have prophase, where there is a tetrad formation and crossing over. We have the homologous pairs lining up for metaphase 1. And then we have a homologous pair separating for anaphase 1. And in meiosis 2, similar things happen, right? We have prophase 1. Basically, we're just getting ready for metaphase. Metaphase 1, we have the chromosomes make a single file line. And notice that each sister chromatid is no longer identical. And then in anaphase 2, we have the sister chromatids being separated. And in telophase 2, we now have 4 cells. And those cells are haploid. They are unique. All of the cells are now different, all 4 of them. And so they're not genetically identical. They are unique, and there are 4 of them, and they are all haploid. All right, good. So the process of meiosis creates gametes with 23 chromosomes that are then going to be able to potentially fertilize one another to create a zygote with a chromosome number of 46, which can then undergo mitosis to make an organism. All right. And there we have it. Okay, we are going to stop there and we will cover this a little bit later. If you have any questions, let me know, and we will finish the rest of meiosis later.